Good afternoon, everyone. My name is Rhiannon Champagne, and today I'm going to be presenting our poster about polydiacetylene's effect on eukaryotic cell growth, viability, and mutagenesis. So the reason that we decided to conduct this experiment is that urinary tract infections are a really bad problem in healthcare settings, and they cost the United States a lot of money each year, not just because of UTIs themselves, but because of their secondary effects or their secondary complications. Also, a lot of the treatments and diagnostic tools that we have for UTIs are not really great. As far as antibiotic resistance goes, there's a lot of trouble with that, and a lot of that is due to the fact that we have very slow diagnostic tools in clinical setting today. So a possible solution that we could come up with is to use a diaper that's embedded with a biosensor that could effectively detect UTI. This particular compound or biosensor that we're aiming to use is called polydiacetylene, or PDA, and it induces a red shift from a blue to a red color in the presence of bacterial metabolites. So what we did in this study is we wanted to make sure that polydiacetylene was not toxic to eukaryotic cells or cells that are closely related to human cells. We used the model S. cerevisiae, or D7 yeast, in order to determine whether it was toxic or not. So the methods that we used were that we put together the PDA liposomes, which are formed by the solvent injection method. We used diacetylene monomers, 10,12-pentacosadionic acid diacetylene monomers specifically. We polymerized them at 254 nanometers for 1, 3, 5, 8, or 10 minutes. Next, we took SRVC and we cultured it with the substance in conical tubes for 24 hours. And so what we did was we took PDA liposomes that were polymerized with UV light for either 1, 5, or 10 minutes, and then we compared them to a positive and negative control. The positive control contained no substance in the tubes, and the negative control contained 300 microliters of 3 molar miconazole, which killed the cells. Now, cell growth was measured by absorbance readings at 600 nanometers for the initial 1, 3, 6, and 24-hour marks. And the methylene blue viability assay was conducted in order to determine the viability of the cells over the 24-hour period. And 3 of these replicates were performed. Student t-tests were also used to determine significance. For mutagenesis assays, we took 100 microliters of the PDA at the different polymerization links mentioned before, and we aliquoted that onto the D7 yeast onto 3 different plates. And we also compared them to a positive and negative control. The positive control was exposed to 10,000 microjoules per centimeter cubed of UV radiation, and the no treatment was used for the negative control. They were incubated for 30 degrees Celsius for 1 day, and then the colonies were observed. So as for results, we polymerized the PDA, and you could definitely see that there was a color shift. When they were first polymerized, they turned blue. We had 3 different stock solutions that we performed in order to, act as replicates. We also exposed all 3 of those stock solutions to 85 to 95 degrees Celsius for 5 minutes, and we induced the color shift, as you can see here. And this is just explaining that we used a blank, which was just DI water. We also had these polymerized for either 1, 3, 5, 8, or 10 minutes, so that's what that means here. Then we also took 2 of the stocks, and we used a 1-to-1 ratio of ammonia to PDA, and that induced a red color shift with the same similar gradient, only a deeper red color. And then we also used a stronger base for stock 2 and 3, and we either added NaOH or 1 normal NaOH in a 1-to-1 ratio or into a 3-to-1 ratio, and that is NaOH to PDA ratio. Then we also took absorbance readings on these particular samples. So the blue PDA, you can see that all 3 of the stock solutions have the same absorbance spectrum. They peaked at about 640 nanometers, and after the redshift, depending on the specific analyte or condition that it was exposed to, you could see that the different stock solutions for red PDA NaOH, it had the least amount of absorbance at 540 to 550 nanometers. And then you had increasing amounts of NaOH that were creating a higher absorbance for the red PDA at those particular nanometer lengths. For the ammonia, the 1% ammonia, you could see an even higher absorbance peaking at these particular wavelengths, and that was most likely due to the deeper red color that you see here. And then our thermochromic trials, where we expose them to 85 to 95 degrees Celsius, they were actually bimodal peaks at 500 and 540 respectively, but it was a very large increase in absorbance. So in addition to this, we did the mutagenesis trials and the yeast viability and growth trials as well. So you can see there that we have 3 replicates of these plates. Our trial 1, trial 2, and trial 3 are A, B, and C respectively. The first plate here in each picture is our positive control, and then the second plate is our negative control. These 3 here are our 1-minute polymerization length, 5-minute polymerization length, and 10-minute polymerization length. And you can tell that in comparison to the positive control, you see a more comparable growth to the negative control. The positive control has a lot less growth in each plate. Now, one thing to mention is that we did see a little bit of background mutation or spontaneous mutation on the 1-minute polymerization length plate, and there were just a couple of very lightly colored red colonies. In addition, we did the yeast viability over time and the yeast growth over time. And as you can see here, as we start out, this is our positive in the blue, our negative in the orange, and then our experimental plates in the gray, green, and light blue bars. The positive control stayed high for viability, as well as did the experimental plates for the most part, and our negative control viability decreased a lot over time, to about 35% by the 24-hour mark. If you look at the gross or the yeast growth over time, you can see that the experimental cultures are following the positive culture, and then the negative control culture is dying off. It never really got up off the ground. Additionally, one other thing to mention is that during one of our trials, our second trial in particular, we did have a positive control that ended up getting compromised. It actually was dumped out a bit during the 6 and the 24-hour trial, but the growth and viability readings didn't seem to change much. We just noted it. As well, here, this little double asterisk, it basically means that our experimental culture for the 10-minute polymerization length ended up not having a similar viability count in the very beginning for our first trial. So overall, we had a lot of very significant time points for viability and growth. Of course, we didn't do any statistics regarding our mutagenesis trials because we didn't really have a very reliable way to count and to quantitatively determine that, but what we did is we just did a qualitative assay. As far as these 2 trials go for viability and growth, we had most of our significance happening in the 6 and 24-hour marks. Sometimes we would have some significance in the 3-hour or even the 1-hour marks, depending on how fast the yeast were growing in the very beginning. There was a little bit of significance, and that could have been due to cohort difference or genetic drift that was just allowing some of them to grow faster in the beginning than others. So in conclusion, PDA was determined to not be toxic. It was polymerized successfully. It was also evident that the stronger the analyte and the more of the analyte that you provided, or the harsher the condition that you expose PDA to, the higher or more significant the color shift was. When PDA was cultured with S-VCA, of course, there was really no apparent toxicity that was determined from our results. Mutagenesis trials seemed to be very similar to, or the experimental plates were very similar to the negative control. And the experimental cultures during the growth and viability trials were very similar to the positive controls, which were supposed to have good growth and viability. So some of the future current work that we're doing on this is we're going to do more growth and viability mutagenesis or soft agar assays on fibroblasts, 2 different cell lines to be exact. And we're also going to be complexing the PDA compound with a hydrogel and then exposing it to different uropathogen metabolites or escape pathogen safe relative metabolites.